Hi, I'm Katie Jones. Welcome to Written Exam 1 Problems. And let's begin. Alright, question number 1. William Bateson and Reginald Punnett wanted to examine the simultaneous inheritance of two characteristics in sweet peas. One characteristic examined was flower color, the other was pollen shape. Bateson and Punnett mated a true-breeding purple flower, long pollen plant to a true-breeding red flower, round pollen plant to generate F1 plants. The F1 plants had purple flowers and long pollen. The F1 plants were allowed to self-fertilize, and the following results were obtained. Purple flowers and long pollen were 222. Purple flowers and round pollen were 14. Red flowers and long pollen were 20. And red flowers and round pollen were 64. So 1A. Propose a null hypothesis to test in a chi-square analysis. And I said that purple flowers are dominant to red flowers, and the long pollen is dominant to short pollen. And here's the Punnett squares that I drew up for that null hypothesis. So then we calculate the chi-square for the data. And so you set it up as such, and you do the math. And you get chi-square being under 68.534, and the degrees of freedom, which is n minus 1, is 3. 1C. So you need to interpret this chi-square value, determine if you should accept or reject the null hypothesis. If you reject the null hypothesis, propose a new hypothesis to test. So we go to the chi-square table, and we find our degrees of freedom and go across. Well, ours was off, so we reject the null hypothesis, and our new null hypothesis that we could test is red flowers are dominant over purple flowers, and long pollen is dominant to round pollen. So number 2. Huntington disease, a neurological disease in humans, is caused by a dominant allele, D. The non-disease allele is small d. These alleles are located on a pair of autosomes. Suppose a heterozygous female, prior to the onset of Huntington disease, marries an unaffected male, and they plan to have a family of 6 children. So here's the Punnett square that I drew up for the 2 parents. So we got half with Huntington disease and half without. Alright, 2A. So what is the probability that all 6 of their children will be afflicted with Huntington disease? So we're going to use binomial expansion, and we set it up like so, and our answer is 0.015625. 2B. What is the probability that 4 children will be unaffected and 2 will be affected with Huntington disease? So again, we're going to use the binomial expansion, and since we use and, we're going to take our 2 answers from our 2 separate binomial expansions and multiply them together to get the probability of 0.0549316406. 2C. What is the probability that the first 3 children will be either all affected with Huntington's disease or all unaffected? So again, we use binomial expansion 2 separate times. But this time, instead of multiplying them together, we're going to add them since the word or is in the problem. And we get 0.04614257001. 3. Propose the most likely mode of inheritance, so either as autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, or Y-linked. For the following pedigrees, affected individuals are shown with filled symbols. Provide a brief explanation for your choice. So we had pedigree charts, but I couldn't get them to work, so we're just going to have to imagine now. So for 3A, I said X-linked dominant, so you need an affected parent to get it. And all daughters of affected fathers have it, and more females are affected. 3B, autosomal dominant. Affects the same number of males and females. At least 50% of offspring are affected and doesn't skip a generation. 3C, autosomal recessive. Skips a generation. Doesn't affect all males, so it cannot be Y-linked. 3D, Y-linked dominant. More females are affecting than males, and all daughters of affected fathers have it. 4. African hedgehogs have 90 chromosomes in a typical diploid somatic cell. So our keyword is a diploid somatic cell. So 90 is the full number, and the somatic cells are regular cells. 4a. How many sets of chromosomes are present per somatic cell in the organism? So we have 90 chromosomes, and we're going to divide it by 2, and that gives us 45 chromosome sets. 4b. How many tetrads are found per cell during prophase 1? 90 chromosomes times 2 will get 180 chromatids, and out of 180 chromatids, we're going to divide it by 4, and we're going to find we have 45 tetrads. 4c. How many sister chromatids are found per cell during metaphase 2? We have 45 chromosomes, divided by 2 equals 22.5 sister chromatids, or supposedly, I guess we could round it to 23, but I chose 22.5. 4d. How many chromosomes are found per cell during prophase 2? It's 45 chromosomes. 5. Consider the following tetrahybrid cross in pea plants, as shown in the problem. Assume that the alleles of all 4 genes assort independently. So I did a test cross because there are more than 3 alleles being shown. So, as pictured. 5a. What is the probability that a dwarf, wrinkled yellow, terminal offspring plant is produced? So, you're going to find each one from your test cross, and you get 1 out of 64. 5b. What is the probability that an offspring plant has the big T, little t, big R, little r, and big Y, big A, little a genotype, or heterozygous for all of them? We pick it all out, and it's 1/16. What's the probability that the first 5 offspring have homozygous, how do I say this, have that genotype? And we're going to multiply all of them together, but it's the first 5, so what we get first is out of 1, and then we have to do out of 5, so that's 1 out of 107374184. And 5d. What is the probability that an offspring plant will have dwarf round yellow axial phenotype or the dwarf wrinkled green terminal phenotype? And so we're going to have to do them separately and then add them together because there's OR in the problem.